Next session is the topic is evolution bites. Why India needs a much improved snake bite therapy. The speaker is Karthik Sunagar. He's from Center for Ecological Studies, Indian Institute of Science, Bangalore. Has taken PhD from University of Porto, Portugal, and guest scientist at Hebrew University of Jerusalem, Israel, and Welcome Trust India Alliance Fellow. Respected sir, please present your topic. Your topic time duration is 15 minutes, and we have 5 minutes discussion. So I hope that it will be a good session for all the participants. Its topic name again I'll announce. It's evolution of bites. Why India needs a much improved snake bite therapy. Welcome, sir.
Thank you so much for the introduction. I hope you can hear me clearly and see my screen. Okay, great. Hi everyone, very good afternoon. This is Karthik Sungar from Center for Ecological Sciences, Indian Institute of Science. I head the Evolutionary Venomics Lab, and today I'll tell you why India needs much improved snakebite therapy. As you all know, India suffers the brunt of snakebite in the world. Over 56,000 people die every single year. And in addition to that, at least 3 times that number suffer permanent disabilities, disfigurements, and amputations. So snakebite in India has a huge socio-economic impact. As you know, snake bites are treated using antivenoms, which are produced by collecting venoms from the snake against which the antivenom needs to be made. This is then injected into horses in sublethal and subtoxic doses. The resultant antibodies are harvested, purified, and sold as antivenom. Studies, including some of our own, have shown that distinct geographic populations of the same species of snake can have very different venom profiles. So, for example, in this species of rattlesnake that we looked at, one of the population was highly neurotoxic, whereas others were highly hemotoxic. So, if one way to make antivenoms using the hemotoxic population, then obviously it will not contain any antibodies against the neurotoxins. Ignoring the geographic variability in snake venoms, we produce our polyvalent antivenom using venoms from a single population in the southern part of the country. This is not the antivenom manufacturer's fault. This is only a venom that's available to them. In India, we also make antivenoms against the so-called big four snakes, which includes the spectacled cobra, the naja naja, Russell's viper, Daboia russelli, common crate, Bungarus ceruleus, and the saw-scaled viper, or Echis carinatus. But there are many other medically important snakes in the country with relatively limited geographic distribution. And last year around this time, we showed that the Indian polyvalent antivenom is not really good at neutralizing envenomations from many of these snakes. So what about the neutralizing capabilities of these antivenoms against our big four snakes? So today in this talk, I'll give you some of the work that we have been doing in the lab, one on naja naja and the other one on Russell's viper. In the interest of time, I'll mostly focus on the results of naja naja. So snake venom research in India basically started with this landmark paper from Professor Gowda's lab in the University of Mysore, where they collected venoms of Russell's viper from 3 distinct locations and subjected them to chromatography and saw regional variations in venom profiles. Since then, many studies have taken place characterizing the venoms of medically important snakes, but a comprehensive analysis of venom compositions and potencies has been lacking. And we also lack the understanding of the influence of biogeography on Indian snake venoms. So if you look at the preclinical effectiveness of Indian antivenoms, again, very limited studies have looked at it in animal experiments. Many studies have indeed looked at the binding efficacies, but as you know, binding does not necessarily reflect on neutralization because the antibodies present in the antivenom may bind to the snake venom toxins, but they may not neutralize the toxic symptoms. So this becomes important to test these in an animal system such as the murine model. Some studies that have taken place on looking at this have followed protocols that deviate from those recommended by WHO. So they are not comparable to each other. You can't even compare them to the outcomes reported by commercial antivenom manufacturers. So to address this knowledge gap, we collected venoms from various locations across the Indian subcontinent, from various biogeographic zones, starting from the coastal plains to the Western Ghats, the deserts, the semi-arid regions, the Deccan Plateau, as well as the Gangetic Plains. We collected venoms from over 128 individual snakes of naja and daboya. and then we subjected them to in-depth proteomic, biochemical, and pharmacological assessments. Then we also looked at the influence of distinct biogeographies on the toxicity profiles of these snakes and the consequent repercussions on the effectiveness of a commercial antivenom. So basically, these two come from these two papers that are currently under review. Before I start my talk, I would like to say that the two projects were largely contributed by Senji, Suyog, and Saurabh from my lab, who were assisted by Vivek and Ashwin. And it also involved some wonderful collaborators in the form of Nick, Jerry, and Ron. So firstly, let's look at the influence of distinct biogeographies on the compositions of venoms. So here I have shown RPHPLC profiles of venoms of cobras from 7 distinct locations across the country. Each peak here is uniquely color-coded. It may contain multiple toxin types. But if you just focus on peak number 4, which we found to be enriched in a form of 3-finger toxin called neurotoxic 3-finger toxin. Basically, this is responsible for all the neurotoxic symptoms that are seen in cobra envenomations. You will see that the area differs drastically between populations. Some populations have really large amounts of this toxin, and others have very limited amounts of it. Similarly, a lot of differences were also seen in the pan-Indian populations of Brussels viper. So we wanted to understand what are these components that are changing between the populations. So we subjected each of those fractions to tandem mass spectrometry, which revealed that the venoms of some populations, like the Eastern and North Indian populations, are almost entirely composed of neurotoxic 3-finger toxins, which is indicated by this blue color here. Whereas some other populations, like the desert population, they have large amounts of cytotoxins or cardiotoxins. So they basically bind to the cardiac muscle and cause cardiac arrest. A lot of differences were also seen in different populations of Brussels viper, basically variations in phospholipase A2s, in QNITs, in serine proteases, and C-type lectins were observed. So now that we know that the venoms vary in terms of their composition, as well as biochemical and pharmacological effects, which I have not shown, we wanted to see what are the effects of these biogeographies on the potencies of venoms. As you know, snake venom composition and potency is largely driven by the ecology of the snake, basically on what they eat. Last year, we showed that the venom of this banded crate, which is a snake specialist, that is, it feeds mostly on other reptiles, has very least potent venom against mammals compared to all other snakes in the Indian subcontinent. So we performed these experiments in the mouse model where different concentrations of venoms are injected to figure out the amount of venom required to kill 50% of the test population using statistics. As you can see, the populations that had really large amounts of neurotoxins were also some of the most potent ones. Whereas the Rajasthan or the desert population, for some reason, was least potent. And this also shows that it also highlights the importance of neurotoxic 3-finger toxins in the ecology of these snakes. A lot of differences were again observed in distinct populations of Brussels vipers, with the North Indian as well as the Central Indian populations being some of the most potent ones. By the way, this also probably indicates that the Rajasthan population perhaps feeds on a lot of other things than mammals. Now let's look at the repercussions of all this variation that we saw on the efficacies of Indian antivenoms. So firstly, we performed venom recognition experiments where on plates, the plates are coated with different venoms. Here, 7 different venoms from 7 different populations of cobras. And then various dilutions of antivenoms are added. So we tested all the major antivenoms that are widely used in the country. And then you measure absorbance. We also add naive IgGs from naive horses, which are never immunized with venom. So you don't expect them to bind. So this is used for calculating the background binding. So basically what you see is blue color here. It illustrates strong binding, whereas reddish to brown color indicates least venom recognition potential. And as you can see, premium serums performs relatively better than all other antivenoms that we tested in the study, immediately followed by Vince, but these two didn't perform so well. And very similar results are also seen against the pan-Indian populations of Brussels viper, with premium serums performing relatively better than all other antivenoms, especially the last two, especially compared to these two antivenoms. What this tells us is that it's very unlikely that these two antivenoms are going to have a lot of neutralizing antibodies because they already lack binding antibodies. But this does not necessarily indicate that premium serums probably works better than all others, because binding antibodies, like I said, may not necessarily reflect on neutralization. So the only way to look at the preclinical effectiveness of antivenoms is by performing something called ED50 experiment, where you take different where you take at least 5 times the LD50 of the venom and mix it with various dilutions of antivenom, and then this venom antivenom mixture is administered into different groups of mice, and then using statistics you can calculate the neutralizing potential. So here in this graph, I have indicated the marketed neutralizing potency in the form of this dotted line. So if you pick up any vial from the market, you will see that it will say it can neutralize 0.6 milligrams of cobra per ml of cobra or Russell's viper venom. So if this peak meets this line, it means that it performs as marketed. So as you can see, it does as marketed against the Southeast Indian population, the Andhra Pradesh population, which is the neighboring population of Tamil Nadu, from where the venoms are sourced for the manufacture of commercial antivenoms. This is also the coastal population, so the venoms probably don't differ so much. So the antivenom performs as expected. But whereas against all other populations, the antivenom lacks efficiency. Particularly concerning was the finding against the Rajasthan population, where every single mouse dies. This is also very interesting because this population had really less potent venom, and this again suggests the importance of cardiotoxins. If there are no neutralizing antibodies against them, then obviously it's not going to do much in neutralizing those envenomation symptoms. Surprisingly, you would expect something similar for the pan-Indian populations of Russell's viper as well, but we actually saw that it performs as marketed against the majority of the populations, barring this North Indian population from Punjab. But you must realize that these tests only look at the abilities of antivenoms in neutralizing the fatal symptoms, and they don't really look at the abilities to neutralize morbidity. So I have summarized the results of the major findings of this study, these two papers, as well as one of the previous papers on this map. Here, the red clouds indicate the snakebite incidence as reported by Suravira et al. So, and I have also indicated the neutralizing potencies as these dotted lines, and the proportion of neutralizing antibodies as these colored solutions for the respective species of snakes. As you can see, the antivenom performs poorly against most of the pan-Indian populations of Naja, and particularly fails against both Naja, Russell's viper, as well as the common crate in Punjab. And of course, it completely fails in Rajasthan. So what we suggest is that India needs to actually make regionally effective antivenoms by sourcing venoms of all the medically important snakes by the region. At least as per me, I don't think producing a single product for the entire country is going to work, because there won't be enough antibodies, neutralizing antibodies against, especially considering the diversity of snakes that we have and the plethora of toxin types they secrete. We have also been sequencing the venom glands of distinct populations, and we already see huge variability in terms of their sequences. So not just the proportion, but the activities are also going to differ significantly. So we propose that we should firstly improve the neutralizing potencies. So the neutralizing potencies of our antivenoms are somewhere here, as I suggested, whereas some of the antivenoms abroad have a neutralizing potency of close to 4 milligrams per ml. Of course, these are monovalent, so they're going to have much more antibodies, but pushing this to somewhere around 2 mg per ml is expected to offer better clinical efficacy. Based on all the studies that are happening in the lab, which hopefully you'll see in the next coming years, we suggest that India needs to make at least 5 distinct cocktails of antivenoms. The amount of neutralizing potencies in each of these Indian antivenoms is really low compared to some of the antivenoms that are manufactured abroad. So that also needs to be improved significantly. Instead of just looking for binding, we should also look at the preclinical efficacies of these antivenoms. And finally, the antivenoms needs to be tested in clinical setting, because that is what will actually tell us whether the antivenoms perform well in humans or not. So with that, I will end my talk here. I would like to thank all the collaborators, all the wonderful students in the lab, all the funding agencies, and the forest departments. Thank you so much for inviting me to give this presentation, and thanks so much for patiently listening.
Thank you, Karthik. We've heard you a couple of times before. Very impressive work. And all of us would agree that you're on the right path, and the studies that you're doing would throw a lot of light on, what really needs to be done, even from the clinical side.
Thank you. Thank you so much.
Two questions for him. There are two questions for you. Yeah, just read it out.
Okay. What is the principal reason for venom variation in the same species within region? Do we find venom variation between juvenile and adult snake of the same species? The question is from Srinivas. Yes.
Yeah, absolutely. So the first reason, like I said, is the ecology and the environment. Venom has been primarily shown to differ based on what the snakes eat. And yeah, there are many studies abroad where people have showed that individuals change their venom profiles over time as they shift diet.
One more question. This is from Jivanjo. Did you estimate the venom yields in different population samples? And in this regard, what is the estimated number of vials for the different antivenoms available in India against Naga?
So we did calculate, but I'm not sure whether there is any direct correlation between the amount of neutralizing antibodies and the number of vials required. So we did calculate it, but we didn't report. And we also calculated the amount of venom that is injected by different populations. Some populations, like the Central Indian population, yield close to, on average, around 400 milligrams of venom in one. I don't know whether they inject all of that into a bite victim, probably not. But at least when you force them to eject their venom into that vial, then they do put out close to 400 milligrams of venom in one bite.
Dr. Karthik, I have one question. Do you have any anti-venom for Humnose pit viper? That is one of the common snake bites we come across. Any plan for making a monovalent anti-venom for this Humnose pit viper?
I would be able to answer that question in a few months down the line. We have been looking at that as well. But yeah, I completely agree that at least in vitro conditions, it doesn't look like it offers any protection. So administering polyvalent antivenom for treating Humnose bite, at least in the preclinical setting, does not seem to be a good idea. But yeah, in a few months' time, I'd be able to answer that much more confidently.
Any more questions, sir? Thank you, Dr. Karthik, for this excellent presentation. We learned a lot of new things about how the anti-venom is produced, what are the differences from various areas of India. Thank you again, once again, for participating in this wonderful conference.
Thank you so much.